Hi, everyone. Welcome to the BGC webinar for the E3SM project. Our speaker today is Bill Riley from Berkeley National Lab, and he'll be speaking on the topic weaker land climate feedbacks from nutrient uptake during photosynthesis in active periods. So I'd like to welcome Bill and thank everybody who has called in, and hand it over to you, Bill.
Thanks, Susanna. And just to confirm, can you hear me and is the presentation up?
Yes.
Okay, great. So as Susanna mentioned, I'm going to talk about this recent paper that came out talking about or discussing how plant activity during periods with no photosynthesis affects nutrient acquisition and therefore the carbon cycle. And this work was done with Qingzhu and Jingyun Tang. And I should also mention it's using the ELM v1 ECA version of the model. And that work was done with several collaborators at Oak Ridge and at UC Irvine. And I'll come to that later in the talk. And that figure there is one of the answers that we're going to get to eventually, but without any color bars. So it leaves that mystery until later in the talk. So here's what I wanted to discuss today. First of all, Susanna mentioned there's a bunch of us that are land modelers in this CBGC group, and then there's others that are not. So I thought it would be valuable to at least introduce the global carbon cycle, why nutrients are important in those cycles, talk a little bit about plant and microbial dynamics and nutrient competition, because that's what this paper focused on, is that the role of that competition leading to plant acquisition, and then follows on to the carbon cycle. And then I'll talk about observations, which have been going on for decades at this point, of plant nutrient uptake in the absence of photosynthesis, and how that motivated the development of the model. And then I'll talk about one of the reasons I think this is important is that the CMIP class of models that are going to participate in the next assessment, the CMIP 6 models, all apply the relative demand approach, as far as I could tell from a review of the literature when this paper was being submitted at the end of last year. And that approach has important implications for this nutrient acquisition question. And then I'll talk a little bit about some of the more mechanistic understanding of how plants acquire nutrients based on enzyme kinetics and how we integrated them in the ELM v1 ECA model. And then talk about the results and implications of those reactions. Okay, so let's start with the background on the carbon cycle. So this is, it's actually a little bit out of date now, but it'll give us a sense of the size of the fluxes, which is what I really wanted to communicate with this figure. This is from the 4th assessment, and this is the whole global carbon cycle. So let me focus on the land, and I'll blow up that part here. So these are the major land fluxes, and really this is just to give you some intuition about the size of the fluxes and why we care about nutrient constraints. So if you look at the purple boxes, which I've added, those are the major fluxes we care about. As I said, they're a little bit out of date. So the fossil fuel flux, I'm hoping you can see my cursor there, is now closer to 9 petagrams of carbon per year, and the land use change flux is estimated this will be around 1, maybe a little bit different than that, but ballpark. So you can think of the sum of those as around 10 petagrams of carbon per year, compared to the gross fluxes into and out of terrestrial ecosystems, which are at least 10 times as large as that. So the argument here is that a relatively small bias in these gross terrestrial fluxes can swamp the perturbation flux that humans are having to the atmosphere. So we have to get these gross fluxes accurately in order to assess the effect of that perturbation that humans are putting into the system. And that's hard. As you probably remember from the last assessment of these models, there was a wide range of predictions of the terrestrial carbon fluxes. And it's hard for a number of reasons. Least among them, I shouldn't say least among them is the fact that there's a lot of temporal variability and a lot of spatial heterogeneity. So I have some just pretty pictures of landscapes to give you a sense of what we're trying to model here. There are, even at sort of site level systems, a lot of heterogeneity in the type of vegetation, There's spatial heterogeneity way below the grid cell level that we try to represent in these large-scale models. There's variability in the hydrology and in the topography and in the temperature. There's a lot of variability in the type of landscapes. This is polygonal tundra, which I'll come back to because it's one of the sites we used to test the model up in Barrow, where the NG Arctic site is working. There's heterogeneity, as I mentioned, in topography and in canopies, multiple layers of the canopies and different types of plants occupying different parts of the canopy. There's larger scale interactions with streams and rivers and estuaries, and sort of this whole issue of climate affecting the terrestrial biosphere leads to really large differences across the landscape. But just as important are the processes below ground, in particular for the question that I'm going to address today, both in temporal variability and heterogeneity, because the acquisition of nutrients by plants, of course, occurs below ground primarily, and plants allocate resources below ground to their roots to acquire nutrients for structure and for water, but here we're going to focus on the nutrients. And you can just see that there's just a very wide range of the below ground structure of roots that the land models are trying to model. And obviously we're not going to be able to model this complexity, so we need to come up with model structures that represent functionally what they're doing. And the other part of the problem, of course, is that the microbial populations that are sometimes facilitating and sometimes directly competing with plants for resources are also extremely heterogeneous. And even if you sort of wash out the details of species level differences between microbes, there's a large variety of functional diversity in soils. And so again, if we're going to try to represent these constraints, we need to think about ways to simplify this complexity. And the other thing that's really complicated about the terrestrial systems are the wide range of timescales that the models have to account for. And I've just, this is just a schematic, but just to give you an idea, at the very fine scale, time scale, sort of hours to maybe a few days to a week, the important processes with respect to this question anyway are this subsurface competition for nutrients that occurs every second. Most of these models work at half-hour to an hour timescales. So at every half-hour to an hour timescale, this competition needs to be resolved in the models and is resolved one way or another in the models. And I've just shown this schematic to give you the sense that at this point, the models are representing several functional groups of microbes. I have nitrifiers, denitrifiers, and heterotrophic decomposers here. Some of the models represent other functional groups too. Roots are actively acquiring nutrients to the extent they can. And then there's also a bunch of abiotic processes which compete for nutrients. And here I'm just representing mineral surface interactions with phosphate or phosphorus molecules for that. And then again, there are multiple substrates that the organisms and the abiotic processes are competing for. Then there's the processes that happen at slightly longer time scales, which include, importantly for the questions here, the dynamic allocation. So most plants will respond to stresses by reallocating their resources. So for example, and there are plenty of observations to support this, if there is a nitrogen limitation, plants that have fungal symbionts that fix nitrogen from the atmosphere will allocate resources to those organisms to help them get nitrogen. The complication, of course, is that that allocation is carbon, but it also may require other macronutrients like phosphorus. So to alleviate a nitrogen constraint, they might have to invest phosphorus, for example. The model I'm going to talk about today has both of these process representations for both of these sets of processes. The successional dynamics, which are also really important in terrestrial ecosystems, refer to things like what happens to these systems after a disturbance. And from just walking around, if there's a disturbance, the vegetation that starts after that disturbance can be very different than the vegetation that was there before, if there was some sort of equilibrium established before that. And those plants that occur in the early successional stages often have very different traits than the climax community. And so this is all just to say, I'm not going to talk about this because the model isn't ready to do so, but this is the FATES part of the ELM work that's ongoing now. Okay, so with respect to nutrients, why do we care? And I just have a little list here, which is probably incomplete, but it'll give you a sense that nutrients in the real world and to the extent possible in the models affect many of the processes important for the carbon cycle. So direct controls over photosynthesis and growth, direct controls over microbial dynamics, including fixation, turnover, and these symbiotic or asymbiotic fungal associations in the mycorrhizal, in the rhizosphere. Nutrients affect allocation. As I mentioned, these, they're living organisms that will reallocate resources given a certain stress. And of course, we care about the losses, partly because there's a radiative component for N2O, and also because nitrogen is a fertilizer, basically, and if you move it from the terrestrial ecosystem into aquatic waterways and estuaries, you can have a response there. And that nitrogen, of course, that's lost can no longer facilitate photosynthesis and carbon uptake from the atmosphere. So I want to spend a little bit of time now talking about what we know from observations about how nitrogen and phosphorus affect ecosystems. And there's information from the FACE studies, but I'll focus just on some fertilization experiment results here that have been synthesized into meta-analyses just to give you some intuition about the size of this effect. And so these types of experiments where people go out into different systems and add fertilizer, nitrogen, phosphorus, and other compounds have been going on for a long time, long enough that there are now meta-analyses of the effects. And so here's one example from about a decade ago, synthesizing 126 different experiments around the world. You can see where the sites are, obviously biased to Europe and North America, but nevertheless, coming up with and these response ratios are for growth, for NPP, net primary production, from the fertilization. And you can see the effect size ranges from, maybe 10% in deserts, not surprising, not so great there, to more than a factor of 2 in some soils, with their assessment overall of the effect is about 29% effect on NPP. Here's a study, another meta-analysis of many experiments, but here they focused on co-limitation, so adding nitrogen and phosphorus separately, and then adding nitrogen and phosphorus. And they also, this paper is interesting because it also synthesized or performed a meta-analysis from marine systems, which might be of interest to other people in the group. I just pulled out the terrestrial part here. Again, the effect size for individual perturbations are ballpark 25% or so. Interestingly, there's a synergistic effect on nitrogen and phosphorus, and that occurs because most ecosystems are not simply limited by one or the other macronutrient. They're typically co-limited. The other thing that can happen, as I mentioned, is that fertilization with one compound can relieve the nutrient constraint of another compound. So, for example, in nitrogen fixation, the enzymes that are required to do that require a lot of phosphorus. So if you add phosphorus to a system that's nitrogen limited, you could alleviate that nitrogen limitation with the phosphorus. And so you get sort of this synergistic benefit. Okay, so it's pretty well established that systems are nitrogen limited, and you have a sense now of the size of those limitations. People have then taken that understanding to what have been carbon-only assessments of the global potential for land to acquire carbon. And so this is an example from Bruce Hungate's paper in 2003 using the third assessment models. And I just want to make the main point he was trying to make here is that if you look at the additional carbon stored over the 21st century, which is the x-axis in these models, you see the values. It's a lot of carbon. That storage required nitrogen, but the models didn't represent that nitrogen constraint. So what he's plotted on the y-axis is an estimate of the nitrogen that would have been required to sequester this much carbon. And then the green lines here show what's actually possible in the global land system. And you can see that it's far below what the models imply would be required, meaning that the land system was nowhere near as effective, would not be nearly as effective as these models suggested. And the same problem existed in the CMIP-5 models, which are shown here. This is a paper from Zonka looking at the CMIP-5 models and doing a similar analysis where he took the carbon-only models in the CMIP-5 and made an assessment of what would these models have predicted if they had represent nutrient constraints. And you can see that between the red and the blue bars for these different RCP scenarios, very large reductions in the capacity. And then Will Weider had a paper in the same year doing an analogous thing, but here and the units are different here because he's comparing net primary production in pp, which is per year growth, between the carbon-only models in gray, and again an assessment, because the models didn't do this, of the nutrient constraints, about cutting it in half with nitrogen constraints, and then again in half if you added phosphorus constraints. So, and that has motivated, many modeling groups for the CMIP-6 exercise to represent nutrient constraints. So now let me shift a little bit and talk about the observations of that we know just from site level observations of what's going on at night in the non-growing season. And the context here is that, as you'll see, these are very large proportions of the nutrient acquisition by plants. And I'll make the point later that the CMIP-6 models largely do not include it. And there's a lot of numbers. This is just a first example from a paper by Josh Schimmel in a grassland in California. And I don't expect you to know these numbers. I'll make a bar chart of this in a second and show you, because we use it for a benchmark for the model. But the conclusion from this study was that about half of the nitrogen that these plants acquired occurred at night. The other interesting thing that they showed is that at night, the plants are much more effective at getting nitrate than ammonium. And that's probably because nitrate is more mobile. During the day, there's a lot more nutrient or nitrate movement with the transpiration flux. And at night, it's all probably diffusion constrained, but it's still much more mobile than ammonium. Here's a figure where, so there are a bunch of papers that have come out over the past several decades where people used isotopic tracers at much finer timescales. And so this is an example of what is often found. These are just two different types of plants. So the daytime, you can see the markers for light and dark. Daytime happens at 9 a.m. here, and you can see over the course of the day an increase in the uptake in both plants of nitrate. And then when it becomes dark, you can see a decrease in this plant and sort of a leveling off in this plant. The important point being that the plants are no longer photosynthesizing at night, of course, but they're still acquiring a lot of nutrients into their roots. And this is just another example, again, where the nighttime fluxes into the plant. This is from the 80s, so these experiments were done in the 70s, where a lot of the nitrogen is acquired in the absence of photosynthesis. And so I was able to find about 20 of these studies where they had measured explicitly daytime and nighttime fluxes. And it was across the board important, somewhere between 30 and 60% of the total uptake occurred at night. And there were no studies that contradicted this finding. So it's pretty clear that these plants are all doing it. Now, one caveat here is that I was unable to find any studies in trees. And so we mentioned this in the paper as a real limitation. I expect that trees are doing the exact same thing because they have all the same types of infrastructure. They have the fine roots and they have the transporters on their roots that are acquiring during the day. There's no reason to think they wouldn't at night, but there was no direct observational confirmation of that. Okay, so the other axis here is the non-growing season dynamics, which turn out to be really important for high latitudes and high altitudes, so cold systems. And in these systems, we know that they're often very nitrogen limited. And from work citing here a paper by Colleen Iverson, who synthesized information about roots, and in the tundra, plants allocate a huge amount of their resources below ground. Up to 90% of the biomass is allocated below ground, probably because they need to get at rare or nitrogen that is rare. We also know that activity in these systems of the roots below ground continues well past when the above ground components of the plants are active. So this is a figure from Bloom-Wary, and actually I just picked out one of the systems. She looked at a bunch of different high latitude systems. It's just to give you a sense that sometimes in these systems you can have the root activity extending months after the above ground plants have senesced. And that was pretty common across all the systems she looked at. And so conceptually, you know, the idea is here, the plants have invested that resource into the below ground, and there's no reason to think it wouldn't be active given the nutrient constraints that they're under, that they would not be trying to get at the nitrogen and phosphorus. We also know, so from direct observations, that this is occurring. This is just one example from Kipper et al. And in this study, they took ammonium nitrate, labeled it with N15, and put it right at the permafrost boundary. This is in Sweden at the Abisko site. So right at the permafrost boundary after the plants had senesced, it was in September or late September, and then harvested the roots of those plants right at the beginning of the next growing season. And what they found is that over the winter, the deep-rooted plants, which are the two that I've pointed to here with the red arrows, acquired much more of the tracer than did the shallow-rooted plants, indicating that those plants did get, now this is not quantitative, so they couldn't tell how many milligrams of nitrogen itself were acquired, but that these plants were getting at it. And this paper was interesting to me because they were very explicitly criticizing a paper that we had written, Charlie Coben led, talking about the role of this nitrogen that's made available toward the end of the growing season. And we used CLM 4.5, which is a relative demand-based model, and therefore was unable to acquire that nitrogen. And here they're showing that it's happening, and I think they're probably right. The other data set I wanted to show you is from the Barrow site, and this is actually from measurements that Terry Chapin made in the 70s. It's at the same site right by where the NG Arctic experiment is going on now, and I've plotted here the measurements he made in red, so you can see the very short growing season. The red is the uptake flux of phosphorus. The brown arrow indicates when the plants actually started to senesce, but the continued uptake well into the later season, well after the plants were no longer active. And the reason that can occur there is shown here. And so this is just a plot from a model. This is a different model than ELM. This is EcoSys, but just to make the point that you're looking at a depth from 0 to about 60 centimeters over 1 year. And this blue zone is the ice-free zone. So there's no ice there and the temperatures are above 0. But that zone extends well into the winter time. So there's a volume of soil there that's above 0 degrees where the plants and the microbes are still active and are acquiring nutrients, even though the above ground parts of the plants are long senesced. So again, here there are fewer, unfortunately, fewer observations, but there are about 10 isotope labeling studies. There was a relatively wide range in the relative effect size. It's hard to do these experiments because it's hard to actually be up there in the winter to make these measurements. But ballpark, the smallest number that anybody reported was about 10% of the annual uptake occurring during the non-growing season. Many of them were higher, up to 50% of the annual uptake occurring outside of the plant growing season. And ballpark, you know, the number I keep in my head is sort of 25 to 30% on average across them. And there were no studies that contradicted this finding. All of them found some uptake that was, you know, of a reasonable magnitude. Okay, so that's the context from the observations. These dynamics are clearly happening in the real world. Let's talk a little bit about how the models represent this competition. And just to give you a schematic here, this is from Ching's paper where he was discussing some of these issues. The concept is you have this complex below ground structure of roots and multiple microbial functional groups, multiple types of substrates that the various organisms are going for, as well as abiotic interactions with these are little rocks, and competition with transport. So you have leaching and gas losses, all of which are competing for these various nutrients, as shown in that schematic. Okay, so how are these models doing this? At the time that I finished this paper, which was just, maybe it was even in January, I found about 12 of the models that were planning to participate in CMIP6 had described their nutrient and carbon cycle components. Every one of them used the relative demand concept. And the relative demand concept is this idea that you first calculate the potential carbon transformation rate, ignoring nutrient constraints. So that's the photosynthesis rate, for example, or the below ground decomposition rate of soil organic matter. You ignore the nutrients at first, then you examine how much nutrients are available. And typically these models use the soil nitrogen, mineral nitrogen pools to do this. Even though they're trying to limit photosynthesis, which is occurring in the leaf, they're limiting it by the amount of resource in the soil. And if there's more demand between the plant and the soil microbial community than exists, they scale the acquisition in that time step relative to their demand. So it's a simple approach that's easily implemented, and all groups, as far as I could tell, in this scale of model, have implemented it. With respect to the issue at hand, though, if you use this sort of relative demand approach at an instantaneous level, which is what most of them do, you cannot have plant uptake in the non-growing season at nighttime because there is no plant demand at that time. It's zero. So what we wanted to do was to develop a model structure that allowed for that to occur. That was one of the reasons we wanted to do this. And the concept goes back to over 100 years now, where Michaelis and Menton did some experiments with microbial populations and here they were looking at carbon, and they found a saturating isotherm, which I'll show you in a second. But what I want to show here is the work of Briggs and Haldane in 1925 that conceptualized the problem in the way we're going to conceptualize the problem for ELM, which is that there's a substrate of interest to the consumer. There's an enzyme that facilitates production or creation of a complex, and that complex it's conceptualized to be in a quasi-steady state equilibrium with some potential and some rate of production of a product and an enzyme being regenerated. And so with this conceptualization, they were able to show the observed Michaelis-Menten response. So what we want to do here is, of course, not to represent each enzyme on the planet, which would be just completely impossible, but to develop a functional form for these interactions that matches the theoretical understanding of them. And almost all of the biogeochemical reactions that I'm going to discuss are enzymatically driven, either by transporter enzymes on the surface of roots or enzymes that microbes produce extracellularly or on their surfaces for transport into the cell. Now, of course, there's also acid-base chemistry and redox chemistry, which are important, which can be important, but with respect to the nutrient acquisition, they're all enzymatically driven. And so this is the equation that is sort of everybody knows about for Michaelis-Menten kinetics, and explained by the Briggs and Haldane, that there's basically this saturating term on a maximum rate that depends on this affinity coefficient or half saturation coefficient Ks. Now, people have been studying this. It's been around for 100 years, and it turns out it doesn't always work. There's been a bunch of studies showing that under certain circumstances it doesn't work, including by our group. And there have been a number of modifications, and I'm showing this one because functionally it's going to look a lot like what I'll show you with our development in a second. And here they recognize that you also need to account for the enzyme concentrations in the competitive network. Okay, so with that background, what we wanted to do was to extend these ideas to the more complex situation that happens in soils, which is that there are multiple substrates and multiple consumers. And so Jin-Yoon, when he came to Berkeley, took this on, and his idea was to expand on the Briggs and Haldane concept, but acknowledge this complexity of substrates, multiple substrates and consumers. And I won't go through all the reasons or the reasons we get the relationship that we do, but with some assumptions that are similar to those that are made in the original single substrate, single consumer representations, we can generate the equilibrium chemistry approximation. So I should mention also, one can solve these equations in all their gory detail with the full kinetics. But that's super expensive and probably impractical for any of the modeling that we do. So Jinyun's goal here was to come up with a conceptualization that could be used like the Michaelis-Menten kinetics. And you'll see the functional form here looks a little bit like the Michaelis-Menten kinetics with some other terms associated with the enzymes produced by the various consumers. And as an aside, I'll just mention Jinyun has been active in the years since thinking about how to use this concept for representative soil control volume. So not representing each individual reaction, but coming up with ways to represent the mechanisms known to exist in the soil through these, well, in this case, through manipulations of these affinity coefficients, both for inhibitory mechanisms, diffusion limitations, which are really important for soil water control on decomposition, and for integrating various microbial traits as we get to more and more complex microbial models with different types of functional groups. And this idea then was extended by Qing when he came here to include nitrogen and phosphorus, and he looked at it, to show you this quickly, looked at applications of this concept at various sites, and here's examples across a coronal sequence in Hawaii, where the experiments were done by other people, but they put in radioactive phosphorus as a tracer and then looked at where it went between being acquired by microbes or in the middle surfaces, and Qing showed very good representation by the model. And he also worked with ammonium in Puerto Rico, and again showed relatively good partitioning. You can see there are some biases here, and the paper describes why we think that was less well represented. But nevertheless, the model is ballpark getting the right partitioning of these nutrients between the various consumers. The other one of the other ones that I'll show here is a site level comparison where there was a lot of data, and this is in an alpine grassland system measured by other people. And what you're looking at here is the ratio of the microbial nitrogen uptake to plant nitrogen uptake as a function of the root biomass density. Here, we're thinking of this as a surrogate for the plant's competitive ability. You can imagine if there are transporters on all of these roots, then it would scale directly. It probably doesn't scale exactly like that, but it's a surrogate for that. And you can see this pattern in the observations. And the blue line is the ECA model with parameters from a different grassland, and then with those parameters modified, not that much actually, but slightly, for the system, can get very good fits. And then we also tested the relative demand approach as it's implemented in the CMIP6 models, and showed that even the functional form is wrong, as well as the other idea that people have, which is not commonly applied anymore, is that the microbes get all of the nitrogen immediately, and then the plants get what's left over after decomposition or mineralization. So the important point here is that these measurements are well represented by the ECA approach, and that no calibration for these other approaches were able to get the right patterns. And I should mention also that this concept, and we've applied it in ELM version 1, it's also been now applied in 2 other large-scale models, ED2 by David Medviji, and in the Orchidea model, because I think other groups are starting to recognize this issue. All right, so let me then describe a little bit about the model, and then we'll get into the results. So this is the ELM V1 ECA model. The paper is in revision right now, and there are several things that are probably important for nutrient competition and nutrient constraints in the model, but I'm going to focus on the first, which is this nutrient competition mechanism. The model also allows for dynamic plant allocation. I mentioned earlier that that's really important, and for dynamic plant stoichiometry. That's another feature of observations, that plants have some flexibility in their stoichiometric ratios in various organs. And Qing developed this based on a very large meta-analysis of stoichiometry and various plant functional types. Okay, so let's get to the results and implications of including these processes. So this is, first of all, just a couple of site-level model results from ELM v1 to give you some intuition about the time course of these predictions. So this is actually from the grid cell that the Schimmel et al. Paper that I mentioned earlier in that grassland measured the nighttime and daytime uptake. And what I'm showing here on the green axis, the left axis, y-axis, is the net primary production of the grass over the course of a day in October. This is actually across all the days in October. It's just a diurnal cycle for the month. And the red is the phosphorus uptake, and the black is the nitrogen uptake over time. And you can see this is very typical from the observations also. There's a lag between the nutrient uptake and the NPP. In this case, the grass was water stressed in the middle of the day, so it sort of leveled off. Typically, this would continue up in a non-water stress situation. And then there's this long tail of nutrient acquisition which occurs over the night. And if you compare with the observations from Schimmel et al. With this estimate of the daytime to nighttime partitioning, it's the model does just right spot on. Lower variability across the month, but still the mean is quite good. We also assembled an observational data set of 123 short-term isotopic tracers. So this is a similar sort of approach to the isotopic tracers I showed earlier from 23 different sites, and then ran the model at all of those sites for all those experiments. And again, the model does quite well at predicting the microbial to plant nitrogen uptake. In the median, you'll notice the range is smaller than the observations, and we don't really know why. My guess is that these are 2-degree grid cells. We simply cannot represent the complexity of all the individual sites in the model. But nevertheless, the mean is about right for this ratio. Okay, so the other thing I wanted to show here is now we're moving from the site to the global model, and I'll give you a sense of how the global model is doing first before we talk about the nutrient issues. So this is, I'll just show a couple of figures. This is for the GPP, and these were all generated from the ILAM package, the model benchmarking package. So I'm going from CLM 4.0 for its GPP bias to CLM 4.5, and the score on top is the GPP score from ILAM, which is a complicated score we can go into later about how it's actually calculated, but it includes a bunch of things like the bias and the seasonal cycle and the spatial structure and so forth, all combined to the current model. So again, the model is getting better over time. You can see there are still biases, but they're getting smaller. We also looked at plant biomass, because of course that's an important result of this nutrient competition. Going from CLM 4 to CLM 4.5, there was a slight improvement, but still large biases to better now with a higher score, but still important biases in some of the high latitude forests and in the tropical systems. The other important thing here for benchmarking is the nitrogen losses, because that's what I'm going to come to as one of the major conclusions of this paper. And so we used for that this paper that Ben Houghton wrote in 2015, where he was criticizing CLM 4.5 for its honestly, it's very poor representation of these fluxes. Ching and I responded to that paper with an analysis in ELM v0, which is basically the same as CLM 4.5, and showed that we could improve it dramatically by integrating the ECA approach. And then this figure here is from the current model, showing very good comparisons to that Houghton et al. Data set for the fraction, an estimate of the fraction of the nitrogen loss from these ecosystems via N2O fluxes. Okay, so that gives you some sense that the model is reasonable, both for the global patterns and for the diurnal cycles of uptake. This is the first result from the global estimate of what happens in the non-growing season and nighttime, showing you the fraction of the annual nitrogen taken up during these photosynthesis and active periods. You can see a clear pattern across latitudes, where it's much higher in the low latitudes of tropics and much lower towards the high latitudes. And this is the figure averaging across longitude. And I put these brackets here just to give you some sense that this fraction is dominated by nighttime uptake at the lower latitudes and dominated by non-growing season uptake at the higher latitudes. And the numbers go from 45% down to 20% on average. Very similar pattern for phosphorus uptake, just for the proportion taken up, proportion of the annual uptake that occurs in the absence of photosynthesis. Very large fractions, obviously, ballpark a half, 40%. And so now, of course, you know, the one point here is that all land models are false. They have they lack processes that we know to be important. The question here was, this is one. It's clearly one. You know, we showed from the observations that these mechanisms occur, and the relative demand models cannot represent it. The question is, what are the implications? How important is that problem? And so to get at that, we did several simulations, 2 sets, each with 2 simulations that I'll show here. From the baseline model, we ran it all the way through the transient to 2010, and then we restarted in 2000 and ran 10 years suppressing the photosynthesis inactive period uptake. And then we basically created a completely different model by turning off photosynthesis inactive period nitrogen and phosphorus uptake during spin-up. So it's a completely different global carbon cycle. It's actually not, it's interestingly not that much different, but it's different. And then ran that all the way through to 2010, and then restarted in 2000 and allowed for this non-growing season and nighttime uptake. And so the comparison over those 10 years gives us a sense of how important these non-growing season and nighttime uptake fluxes are. And here's the result for N2O. I'll show you leaching in a second. You can see, so this is the difference between the simulation that allowed and did not allow uptake during those periods for the N2O emissions. And the concept here basically is that if you allow ecosystems to acquire nitrogen when they're not photosynthesizing, the system is much tighter for nitrogen. There's less mineral nitrogen in the soil, and so there's less available for leaching or gas fluxes, gas losses. You can see where that the differences are largest in the tropics, and although there is some boreal forest increase also, but it's dominated and sort of very focused in the around the equator. The numbers turn out to be large. So this is ballpark. These are the 2 simulations. So think about, you know, basically 6 teragrams of nitrogen per year. The global warming potential of that is large, somewhere around 2.5 to 3 petagrams of CO2 equivalents. If you compare that to the current land sink, now I've converted the units here. I'm doing this for global warming potential, so it's in CO2 equivalents. The current land sink is somewhere 0 to 12, maybe a little bit larger, but ballpark 0 to 12. Varies a lot. There's a lot of inter-annual variability in the land sink. So this N2O flux has, at a minimum, 25% global warming potential of the current land sink, and it can be huge compared to the land sink when that land sink is small, tap like regular. We can also look at the leaching losses, which are even larger in nitrogen. Of course, they don't have a direct radiative effect, although a lot of that nitrogen is moves into the rivers and then out into the estuaries and the ocean and can be denitrified and returned as N2O. But we didn't do that calculation. I think the IPCC calculation is several percent of this leaching loss flux is guessed to come out as N2O. But here the response is more diffuse in space. You can see it's still a lot happening around the lower latitudes. There's a much larger effect at high latitudes. This is almost all happening during snowmelt. So again, the idea being that systems that have plants that can acquire nitrogen and phosphorus during the non-growing season will do so because it benefits them and they have the infrastructure to do so. That makes it the concentrations in the soil lower, so when snowmelt occurs, there's less to be lost as leachate. And you see that pretty clearly across those systems. Okay, and the last thing I wanted to show is just looking down on the North Pole here at the high latitude responses to give you a sense of how important this is for the high latitudes. Many, of course, some of these very high values that are above 40% are in systems that have very low turnover anyway, so it's not that much carbon implications. But across a lot of the boreal forests, you can see 20, 25, 30% of their annual uptake occurs over, this is all non-growing season acquisition. Oh, and the other thing I should mention, which I don't have a figure for here, is that it varies a lot depending on the plant functional type, just the different characterizations of the below ground traits, the root traits affect this acquisition. Okay, so let me just wrap up here with a quick summary. It's pretty clear both from observations and from this model that allows for photosynthesis inactive period uptake that a large fraction of plants' acquisition of nitrogen occurs outside of these periods. 20 to 60% is our estimate across the, individual grid cells across the globe. About half, 45%, if you weight it by NPP, and slightly less for phosphorus. This is the nitrogen number. I think it was 42 or 43% for phosphorus, but still very large fraction occurring when all of the CMIP6 models and the other version of ELMv1 will not allow for it. They just simply can't get it. And if they don't get it, they are biasing their systems to be much leakier in nitrogen. Much higher leaching fluxes and N2O emissions will occur as a result. There's a large, just from the N2O part alone, a large global warming potential associated with that. And although I didn't spend any time here, there's likely a large effect on the carbon cycle. It's a little bit harder to assess. We didn't run these models out to 2100, but these fluxes, sort of 16 to 19 and 6 teragrams nitrogen summed, probably, it's hard to actually say. We need to run the models, but could be responsible for tens of petagrams of carbon differences. So it's a big, 10 to 100 petagrams of carbon is my back of the envelope estimate across the 21st century. So it has a big effect. It would have a big effect also on the carbon cycle. And that's it. Those are the papers that were cited here if you're interested in some of the details.
Okay, great. Thanks a lot, Bill. I'm going to end the recording here, and then we can have some discussion offline or off the recorded part.
Okay.
So, all right.